Hello, welcome to this video on proteins. So we went over carbohydrates in our previous video, and so we will go over the monomers and the polymer form of proteins first. So remember that the monomers of proteins are called amino acids, and amino acids are going to have 20 different types. Here's the basic structure of amino acids right here. So all amino acids have a amino group, NH2, and a carboxyl group, right here, C double bonded to an O, and a hydroxyl group, a central carbon, a hydrogen, and right here, bonded to the central carbon is the side chain, or the R group, which is going to have or be a certain group that determines the type of amino acid that we have. And remember, when we link several amino acids or monomers together, right, the polymer form of proteins are called polypeptides. And right here, we have an example of a polypeptide. We have the amino acid tyrosine right here and the amino acid glycine. And you can see that they are both bonded by a peptide bond right here between the N of the amino group from lysine and the C of the carboxyl group from tyrosine. And you can see that the amino acids are different because they have different side chains right here, right? So let's look at the 20 different types of amino acids, right? So there are several right here, and I color-coded them helpfully. The orange being nonpolar amino acids, and the blue being the polar amino acids, right? And depending on the side chain, right, the amino acids have different properties, right? Nonpolar or polar or polarity being one of them. Next, we'll talk about the levels of protein structure, right? So we start in the beginning with the primary protein structure. So the primary protein structure is just one polypeptide in a straight chain, right? So we can see the amino acids right here represented as circles, and we have the abbreviation right here. And the amino acids are bonded together through peptide bonds, right? It's just a straight chain. Next, we have the secondary level or structure of proteins, right? So we have one shape called the alpha helix, right? And the reason why the amino acid or the polypeptides are able to give you that helical shape is because of the interaction, the hydrogen bond interaction between the groups of the amino acids, not the side chains, but the amino and the carboxyl groups. And that the hydrogen bonds are represented by the blue dashed lines right here. The second type of secondary structure shape is called the beta pleated sheets, right? So you can also see right here the hydrogen bonds between the those groups and the amino acids are going to give you this kind of folded kind of sheet shape called the beta pleated sheets. Moving on, we go to the tertiary structure, right? So the tertiary structure is going to be giving you more of a three-dimensional shape, and this is caused by more folding, right, of the polypeptide, and that's caused by side chain interactions, right, the R groups, right, between the amino acids. So as you can see right here, the purple line right here, right, is just going to be a bunch of amino acids bonded together through peptide bonds, right, and we can see how the side chains, right, of amino acids are interacting among one another, right? So we have an amino acid right here. The methyl groups are interacting with the methyl groups of another amino acid, right? The same amino acids right here. So in this case, this is called a van der Waals interaction, right? Where nonpolar, right, groups are interacting with one another, causing attraction. We have an ionic bond between two amino acids right here, one amino acid here and another amino acid here, right? The negative and the positive side chains are going to be attracted together through an electrostatic attraction. We have a disulfide bridge. Sulfur is going to be covalently bonded to each other between two side chains, between two amino acids, and we have a hydrogen bond between two side chains from two different amino acids. Okay, so what does this result in, right? A kind of a 3D shape, right? Because of the side chain interactions. Finally, we get to the fourth structure, the quaternary structure of protein. So this is where we have more than one polypeptide, right? So from the primary, secondary, and tertiary structures, we only had one polypeptide. But in the quaternary structure, we have more than one polypeptide and several polypeptides that are going to interact with one another through side chain interactions. And this is going to produce basically several subunits of polypeptides or proteins. So an example of a quaternary structure is collagen, right? We have 3 different polypeptide subunits, right? Each color-coded, forming one whole protein structure. We have another example right here, hemoglobin, right, which has 4 subunits, 2 different types of subunits, the alpha subunits and the beta subunits that are going to also interact with one another through side chains and form one hemoglobin protein. Okay, so this was just a quick review of the protein macromolecule. Hope you enjoyed. Please like and subscribe and follow for more content. Thank you.